it's Gabrielle, and this video is a sample from Studyclix Boost, our new rapid revision tool. Go to studyclix.ie/boost to find out more.
In this video, we will investigate the production of alcohol by yeast and then test for the presence of the alcohol that's produced. Alcohol is produced in industrial fermentation on a large scale to meet consumer demand. This practical on your Leaving Cert syllabus shows the basic process behind alcohol fermentation. We will use yeast to ferment the monosaccharide glucose into carbon dioxide and ethanol. Now, this practical is carried out under anaerobic conditions, meaning no oxygen is present. In the second part then of the practical, we'll carry out a test to show the presence of the ethanol that is produced. So to prepare the alcohol, in this case we're preparing ethanol, we mix the glucose and water in a conical flask. We boil the solution for 5 minutes to remove the oxygen and thereby creating anaerobic conditions. We cool to 30 degrees Celsius and add yeast. We then cover the solution in the flask with oil, again to keep the oxygen out. We then place a fermentation lock into the conical flask. We place the conical flask in a thermostatically controlled water bath at about 30 degrees Celsius and we leave it overnight. So what we notice then is carbon dioxide is produced by the yeast during fermentation. You will notice foam produced inside the conical flask. Now, just one or two little notes to point out to you here. The first one is that this airlock, it allows the release of carbon dioxide, but it prevents microorganisms from entering. And if we wanted to, we could place lime water inside the airlock. That lime water would then turn milky in the presence of carbon dioxide. The second thing to note is that you might be asked in the Leaving Cert exam, how did you know when fermentation had finished? Well, the answer to that is no more carbon dioxide gas is produced, so no more bubbles can be seen. After then 24 to 48 hours, we test for the presence of the other product, which is ethanol. So to do this, we filter the contents of the conical flask, we transfer 3 centimeters cubed of the filtrate into a test tube, and then add 3 centimeters cubed of potassium iodide solution and 5 centimeters cubed of sodium hypochlorite solution. You will notice that the solution then turns a brown-orange colour. Now, be sure you're able to spell these chemicals correctly and that you can state the starting colour, which is a brown-orange colour, and the finishing colour. So when we place this test tube into a water bath at about 60 degrees Celsius for 5 minutes, if pale yellow crystals form, well then ethanol is present. So the result is that ethanol was present in the conical flask containing the yeast. Controls are used in experiments for comparison purposes. As a control, we used the exact same diagram as the first diagram, but without any yeast. And in this case, no carbon dioxide or ethanol is produced. Before we started this practical, we made sure that all of our glassware was sterile. This again is to prevent any contamination by other microorganisms. We also wore gloves and safety glasses during the practical, as both the sodium hypochlorite and the potassium iodide are irritants.